Hello everyone, welcome to the interdisciplinary CDE program conducted by the Department of Microbiology. I'm Mareena Mathew. The topic for today's session is Superbugs, a global threat. I hope many of you are familiar with this scientist, Sir Alexander Fleming, the man behind the miracle drug penicillin. He discovered penicillin in the year 1928. And he noticed that the mold growing in his lab can kill some of the bacteria. At that time, it was titled as the miracle drug that saves lives. Though it was discovered in 1928, it was found successful only in laboratory animal. Actually, researchers and scientists were not sure whether that can be safely applied to human beings. Then started Second World War in 1940s. Many soldiers were dying of wound infections. One of the co-workers in penicillin research wing, Dr. Flory, took this penicillin to the war zone and gave it to soldiers who were dying of infections. And that saved the lives of many soldiers. And can see the title, Thanks to Penicillin, He'll Come Back Home Soon. So that saved the lives of many soldiers, and that is titled as the miracle drug. The researchers who are behind this, Dr. Flory, Dr. Chain, and Sir Alexander Fleming, was awarded Nobel Prize in the year 1945 for the discovery of penicillin and its curative effects against various infectious diseases. But the man behind this miracle drug, before leaving this world, gave us a warning. An ignorant man may underdose himself, make them resistant, rendering penicillin ineffective in the future. And in 2003, the former CDC director, Dr. Tom Frieden, stated that the more we look at drug resistance, the more concerned we are. And it shows us that the end of the road is not very far away for antibiotics. Yes, there started reports coming about drug-resistant bacteria in early 1990s. As always, everyone thought that this is going to happen in very few people. This is not something very dangerous. But there started reports coming again and again. And is our topic for the day, superbugs. The strains of bacteria, viruses, parasites and fungi that are resistant to most of the antibiotics and other medications commonly used to treat these infections caused by them is known as superbugs. This is the in vitro analysis in a microbiology lab of antibiotic sensitivity. In the culture plate on the left hand of this technician, you can see many zones of inhibition around the antibiotic disc. And on this hand, on his right hand, this is what we are facing now, the present situation. Only few antibiotics are showing the zones of inhibition, and that too very less in diameter. So this is a picture of antibiotic sensitivity test, past and present. Let's have a glance at the mechanisms of drug resistance shown by bacteria. It can be intrinsic or acquired. Intrinsic means bacteria, they are already resistant to certain antibiotics. We know that they are resistant to certain antibiotics. And acquired is something bacteria acquires by different means. So the drug resistance shown by bacteria can be intrinsic and acquired. One of the common mechanisms shown by bacteria in drug resistance is target change. That is, bacteria will change the binding site, target site for antibiotics to bind. This is very commonly seen in the case of the antibiotic erythromycin. Bacteria shows resistance to erythromycin commonly by target change. Here you can see it very clearly that usually the target will be like this, and for superbugs the target will change to another format. So the drug cannot come and bind to the site. Another mode of drug resistant mechanism is breakdown, enzymatic breakdown. That is, the bacteria, superbugs, will produce an enzyme. With the help of an enzyme, with the help of that enzyme, this antibiotic is broken down. Very common example is penicillinase enzyme or beta-lactamase enzyme, which is used by bacteria to resist the antibiotic penicillin. Another mechanism for drug resistance is block. That is, the bacteria will not let the antibiotic enter into its cell. So we are taking the antibiotic, but bacteria will not, superbugs will not permit the antibiotic enter into bacterial cell. Commonly seen in the mechanism, this mechanism is commonly seen towards the antibiotic tetracycline. And another mechanism is efflux pump, where the bacteria will take the antibiotic, bacterial cell will accept the antibiotic, but in the case of superbugs, the antibiotics which is already entered into the bacterial cell is very quickly eliminated or pumped out from the bacterial cell. So that is efflux pump, which is a very common mechanism of drug resistance seen in most of the multi-drug resistant bacteria. So these are few examples for drug resistance mechanisms shown by bacteria, and all these mechanisms bacteria acquire by gene transfer. That is, when a common normal bacteria is attacked by antibiotics, superbugs, that is drug resistant bacteria, will come with the gene helping for drug resistance, and the gene can be easily transferred by gene transfer mechanisms like transduction or conjugation to the normal bacteria. So that by this process they can also show the property of drug resistance, or they can also become superbugs. Let's see how fast bacteria are in developing resistance to antibiotics. We'll start from the first antibiotic, penicillin. 1941 was the year year of release of this particular antibiotic, and immediately by 1942 the bacteria Staphylococcus aureus developed resistance to this drug. In 1960 the drug methicillin was introduced against penicillinase resistant bacteria. In the same year itself, Staph aureus developed resistance to methicillin. In 1980, cephalosporin, cefotaxime was released or introduced, and by 1983, Escherichia coli that is extended spectrum beta-lactamase possessing Escherichia coli, developed resistance. The fungi Candida developed resistance in the year 2004 to a drug called caspofungin, which was developed in 2001. Just like that, bacteria is very fast in developing resistance to antibiotics. Whenever they develop a mechanism to resist a particular antibiotic, immediately they'll transfer the resistance mechanism to the next bacteria. So this process of transferring drug resistance among bacteria is happening very fastly. And World Health Organization listed a pathogen list, a priority pathogen list. They will revise it in every 5 years. And this is the present list of priority pathogens according to drug resistance, which is categorized as critical, high, and medium. The critical group includes Acinetobacter baumannii, which is known as a nightmare bacteria, Pseudomonas aeruginosa, and most of the members of Enterobacteriaceae, especially Escherichia coli and Klebsiella. Under the category high, there is Enterococcus, Staphylococcus aureus, Helicobacter, Campylobacter, Salmonella, and Gonococcus. And under the category medium, there is Pneumococcus, Streptococcus pneumoniae, Haemophilus influenzae, and Shigella. WHO. The reports of first superbug in India was published in the Journal of Association of Physicians in India, JAPI, in March 2010. And they named it as New Delhi metallobeta-lactamase bacteria, NDM1 superbug. So that is the first reported superbug in India. Let's see the factors favoring spread of antimicrobial resistance. The very first factor is poor infection control practices in hospitals. The hospital should have a keen interest in infection control practices. The staff hygiene should be properly managed. The sterilization of all the contaminated equipments should be properly done. And if there is a case of superbug infection in any hospitals, that should be reported to the authorities. So the infection control practices in every hospital is playing a key role in favoring or in supporting the spread of antimicrobial resistance. And the second factor is lack of hygiene and poor sanitation. Just like any other infection, microbial infection, the infection caused by superbugs can also spread by the lack of hygiene and poor sanitation. Third is irrational use of antibiotics without following the susceptibility reports. See, under emergency, one can start treatment, but the treatment plan should be cross-checked with the antibiotic susceptibility reports. Uncontrolled sale of antibiotics without prescription, which is the usual practice common with people. Overprescribing of antibiotics and patients' non-compliance. Every patient should be educated about the judicial use of antibiotics, the need of completion of the dosage, the need of, even if they get cured, even if they get relief of symptoms, that antibiotic that should be completed, that course should be completed. For such an advice should be given to every patient when you prescribe the antibiotics. And last but the most important factor is unnecessary antibiotic use in livestock and fish farming. It is said that more than 50% of antibiotic use is happening in livestock industries. And through their fecal matter, we come to the common water source, we come to the soil, and whenever bacteria is getting a chance to learn about the molecule, learn about the antibiotic molecule, and that immediately they will learn how to develop resistance to this particular antibiotic. So what we can do is reduce the chances for getting bacteria a chance to learn about antibiotics. Use antibiotics only when it is necessary, and control the unnecessary antibiotic use, especially in livestock industries. And this is a list showing countries with highest increase of antibiotic consumption. And you can see India is in the highest position. So this is in 2000 and this is in 2015. India is rating high in the consumption of antibiotics. So that is an what are the problems associated with antibiotic resistance? Definitely, that will increase the duration of treatment. The patient may require more days of hospital treatment. The duration of treatment will be more. When the duration is more or when the dose is high, again drug toxicity can be expected. The cost of treatment will be more, and there is chance for morbidity and mortality. Increased chance for morbidity and mortality. So these are the common problems associated with antibiotic resistance that general public face. And this shows the impact of antimicrobial resistance globally. You can see the current pandemic COVID-19, influenza, threat of H1N1, malignancies, and it is said that by 2050, more than 10 million deaths will be there only because of antimicrobial resistance, only because of infections which cannot be controlled by the antibiotics, antimicrobials available. So this shows the actual threat of antimicrobial resistance. So there is a threat of death and at the same time a great economic loss. About 66 trillion pounds will cost, that is a cost that we are expecting by 2050 to manage drug-resistant infections. And the real threat is not this actually. This is the real threat that we are facing. Since 30 years, there is no new class, no new molecule of antibiotics introduced. So, pharmaceutical industries, they're not keen enough to synthesize, to introduce new molecules of antibiotics since 30 years. So bacteria is getting more and more chance to learn about the antibiotic molecule, and we are getting more and more lazy. We are not putting efforts to introduce new molecules of antibiotics to the industry. So that is a real threat in this situation. And what is done globally to, to save the nations, to save the world from this threat? So this Global Antibiotic Research and Development Partnership, which is a joint initiative of WHO and DNDi, Drugs for Neglected Disease Initiative. And this group it actually encourages the research and development through public-private partnership. And by 2025, they are planning to develop at least 5 new treatments, new drugs targeting drug-resistant bacteria identified by WHO. And this is WAV 2021. It is World Antimicrobial Awareness Week, which is observed from 18th to 24th of November in every year. And in this year, the theme is Spread Awareness, Stop Resistance. And the overarching slogan of WAV is Antimicrobials should be handled with care. So there is no time with us to wait. We have to secure the future from antimicrobial drug resistance. As we are securing water and air for next generation, we have to save the antibiotics for our future generations. Thank you.